Welcome to the 11th UC Davis Grad Slam competitions. I am Jean-Pierre Delplanque, Vice Provost-Tendean of Graduate Studies. I'm also a Professor of Mechanical and Aerospace Engineering. I am honored to have all of you joining us at our UC Davis semi-final for Grad Slam. We, Graduate Studies, are so proud to be part of this annual UC-wide competition because it presents us with the opportunity to highlight the tremendous research and scholarship being carried out by our students at UC Davis and learn a few things ourselves in the process. It is research that is poised to truly make the world better, and we need it now more than ever. We have challenged graduate students to condense hundreds of hours of academic research and discovery into just one 3-minute presentation, so good luck with that. That's a remarkable feat, no kidding. And today we'll get to hear from our 10 campus finalists who have excelled in this challenge. Our UC Davis top 10 were selected by a panel of judges from a large and competitive submission pool. And based on the students' presentations today, we will award first, second, and third place prizes, plus 2 special awards: the Public Impact Prize, presented by the UC Davis Public Scholarship and Engagement, which will go to the student who demonstrates how their research has or will have significant impact by addressing a public concern or topic. And the People's Choice Award, which will be selected by our audience at the end of the program, all of you here in person, and also those of you watching on live stream. Each of you here in Walker Hall will vote for this award by scanning a QR code that will be shown on the screen after we're done to access a voting survey at the end of our program. Attendees tuning in virtually will have access to a link that will be posted in the chat of the interface. You will have roughly 15 minutes to cast your vote. The first place winner selected today will go on to compete at the UC system-wide final, which will be held at the UC Student Center and UC Student and Policy Center in Sacramento. That will be on April 29th, so it's coming up pretty quickly. Before I introduce our judges and contestants, I want to thank our Grad Slam sponsors for their generous supports, including the Grad Pathway Institutes for Professional Development, the Office of Research, Public Scholarship and Engagement, and Global Affairs. I'd also like to thank Rinki Sethi, who is not only supporting this competition as a judge for the 3rd year in a row, but also contributed as a sponsor this year. Thank you all for your support of graduate education and professional development. I would like now to take a moment to acknowledge the land on which we are gathered. For thousands of years, this land has been the home of the Patwin people. Today, there are 3 federally recognized Patwin tribes: the Katil-Dehe Band of Wintun Indians of the Colusa Indian Community, the Quetzal-Dehe Wintun Nation, and the Yocha-Dehe Wintun Nation. The Patwin people have remained committed to the stewardship of this land over many centuries. It has been cherished and protected as elders have instructed the young through the generations. We are honored and grateful to be here on their traditional land. It is now my pleasure to introduce our judges for this year. Judges, please wave when I say your name. That'd be great so everybody knows who you are. Dr. Simon Atkinson serves as Vice Chancellor for Research at UC Davis. He previously served as Vice Chancellor for Research at the University of Kansas from 2019 to 2023. In addition to research administration at KU, his portfolio included oversight of 11 research centers and the KU Natural History Museum. Simon is a molecular cell biologist recognized for studies in the cellular mechanism and prevention and treatment of acute kidney injury. He has taught cell biology and biochemistry to undergraduate and graduate students as well as medical students. In 2005, he co-founded Infotron, a life sciences company. Dr. Atkinson earned his PhD in molecular biology from the University of Cambridge and his bachelor's in cell and molecular biology from King's College, London. He was also a postdoctoral fellow at the Johns Hopkins University School of Medicine. Dr. Ann Kimtrump is a research scientist, veterinarian for the California Department of Public Health, Vector-Borne Disease Section. She is a subject matter expert for and reviews and summarizes reported cases of vector-related reportable diseases, including Lyme disease, anaplasmosis, babiosis, tick-borne relapsing fever, rickettsial diseases, hantavirus, and plague. Wow. Dr. Kimtrump helps coordinate environmental follow-up of select vector-borne diseases case in VBDS biologists and other local health agencies. She provides input on public health education material and was the initial coordinator for the California Department of Public Health Lyme Disease Advisory Committee. Dr. Kimtrump regularly gives presentations to a wide variety of audiences, from public groups to medical providers to students. She enjoys mentoring students and interns who volunteer at VBDS. She lives in Davis, California, and attempts to squeeze in bicycle rides between her work, family, and other civic volunteering efforts. Dr. Stacy Muse is a seasoned leader in community engagement and strategic program development, with over 20 years of experience in higher education, nonprofit management, and public service. She currently serves as the Director of Engagement in the Office of Public Scholarship and Engagement. She leads initiatives that bridge university resources with community needs, fostering inclusive and equitable environments. She earned a PhD in higher education from the University of Denver and a master's in nonprofit leadership from the University of San Diego. Dr. Muse's work is deeply rooted in democratic engagement and social justice, ensuring that underrepresented community voices are integral to the decision-making process. Rinki Sethi is the Vice President and Chief Information Security Officer at Bill.com, where she leads the global information technology functions and is also responsible for leading efforts to protect Bill.com's information and technology assets and advises the company's continued innovations in the security space. Rinki served on the board of directors of several companies, including ForgeRock and Voltree. She brings decades of security and technology leadership expertise and was recently VP and CISO at Twitter and Rubrik Incorporated. She earned a bachelor's in computer science engineering from UC Davis and a master's in information security from Capella University. Outside of work, Rinki spends time with the family. She loves to travel, exercise, and watch basketball. It says Go Warriors here. Davis Mayor Bapu Vaitla. Mayor Vaitla was elected to the Davis City Council in November 2022. He works on issues of gender equality with Data 2X, an initiative housed in the United Nations Foundation. He was born in India and grew up in California. He holds a bachelor's in nature and culture and a master's in international agricultural development, both from UC Davis, as well as a PhD in international relations, political economy from Tufts University. He has received a Mickey Leland International Hunger Fellowship, a Fulbright Scholarship, and a postdoctoral fellowship from Harvard University. Dr. Gordon Walker is an award-winning speaker, science communicator, published author, and social media star with over 3 million cumulative followers. He has a PhD in biochemistry and molecular biology from UC Davis, where he studied the microbial ecology of wine fermentations and yeast genetic. He's a passionate advocate for mushrooms, fungal conservation, and environmental sustainability. His new book, Passport to Kingdom Fungi, will transform the way people learn about mushrooms and fungi, serving as the go-to resource for a whole new generation of mycophiles and people interested in foraging, food, and fermentation. Thank you to all of our judges for supporting our students today. Now I'm excited to introduce our 2025 UC Davis Grad Slam Top 10. If you don't mind standing when I call your name, that'd be great. Emma Bernard, PhD in candidate in material science and engineering. Latifa Nassim, PhD candidate in biochemistry, molecular, cellular, and developmental biology. Christian Pratt, PhD candidate in physics. Olivia Feldman, PhD candidate in ecology. Nana Moraes, PhD student in anthropology. Sarah Ali, PhD candidate in integrative pathobiology. Yara Khatib, PhD candidate in pharmacology and toxicology. Jacqueline Lang, PhD student in agricultural and environmental chemistry. Diana Malarczyk, PhD candidate in anthropology. Jordan Crawley, PhD student in linguistics. And I'd like to take a minute to personally thank all our students who submitted an entry for this year's competition. I know our students have so many things to do and going on in their lives. It's a great honor to be able to see, even if it's just for a few minutes, the incredible research that they're doing at UC Davis. At this time, I'd like to invite the first speaker, to present. The order of this year's presentation was decided at random. Please help me welcome Emma Bernard.
Thank you. Take a moment to imagine the vastness of the universe. How many stars do you think are out there? If you guessed something like 200,000 trillion, you'd be right. Now consider this. Do you think more or less data was generated in 2024 alone than the total number of stars? Right now, this number is slightly lower, at 150,000 trillion bytes. To put that in context, that's enough data for each person on Earth to have 125 iPhones. And in the next 5 years, we are on track to continue generating data at this astronomical rate, especially with the advent of artificial intelligence. But to truly understand the implications of all this data, we need to talk about how memory is stored. Lots of our memory is kept on the cloud, but in reality, these are massive data centers worldwide full of servers that store our information as bits of ones and zeros. A sequence of these bits is known as a byte and is the fundamental building block of all memory. The issue is that the way servers store memory right now requires constant energy input and demands vast amounts of water for cooling. In fact, last year, global data centers used nearly the same amount of electricity as the entire United Kingdom, and individual data centers can use enough water per day to fill 8 Olympic-sized swimming pools. So as we generate more data, we use more resources, potentially leading to an energy crisis. So what do? As generating less data is not a viable solution, what we really need is a way to store information without constant energy input. Kind of like words in a book, right? Once you've spent the energy to write the word, it's stable without power. Let me introduce you to the Skyrmion. And no, that's not a Star Wars character. The Skyrmion is a tiny, swirling magnetic object that has the potential to store the ones and zeros of our memory in its magnetic configuration. And these bits, kind of like words in a book, are stable, offering a low-power solution. However, Skyrmions are not used in devices yet because they can be hard to control, limiting the speed at which we can read and write new memory. So with my research, I study the ultra-fast behavior of Skyrmions. To do this, I shoot a laser at the Skyrmions to excite them, and then I track their changes down to a trillionth of a second using X-rays. At these timescales, we are observing the extreme behaviors of Skyrmions so that we can shed light on how we can manipulate them so that future devices will have the speed and capacity of current servers, but with the much needed energy savings to store our ever-expanding universe of data. Thank you.
Stay here.
Oh, sorry.
Can you tell us what's next for your research?
Yeah, next week, actually, I'll be visiting the Advanced Light Source down in Berkeley to image this 3D structure of the skyrmions because, you know, their structure definitely plays a role in their properties.
How big are these?
They can have a huge range depending on the material, but the ones I study can be anywhere from 10 to 100 nanometers.
Okay, right. How did you pick material science as a specialty?
So when I was choosing material science, what really drew me to the field was how it offers tangible ways to solve problems that I care about. Energy and energy efficiency is a big one, and with lots of different aspects of material science can answer these questions, from batteries, solar cells, but then in my case is reimagining how devices can operate.
What do you think we could do to interest high school students in material science? And what I mean by that is making it visible to them.
Yeah, actually, in my group, we work with MESA students every summer. And how I like to explain material science to them is first by just, you know, getting the materials in their hands. You know, it is a very nice, tangible field of research. But I also think that discussing how everything we do is made of some material. And so how can you improve it? Because that has real impacts on efficiency.
Okay, makes sense.
Yeah.
What are your future career goals?
So I think whatever I do, I want to stay working with materials for devices, whether that's memory or any other sort of device. But I'd like to take it from an academic approach long term, either at a national lab or in academia.
Mm-hmm. Okay. I'm going back to my question about visibility of material science. How did you get into it, right? What brought you to it?
So it's actually funny. I was first interested in material science because I wanted to work on biopolymers. Okay. I really wanted to be able to recycle the plastics that we use every day, and I actually worked for a company that made bioplastics, so polylactic acid, and it was a great experience, and I felt like there's kind of a, you know, lot that can be done there. But when I was in those classes to study biopolymers, I was exposed to all the other materials that have such a big impact on everything we do. And I got really interested in functional materials.
Okay, that makes sense. Thank you.
Yeah, thank you.
Our next speaker is Natifat Nassim.
Picture your morning commute. There's cars everywhere, you're stuck in traffic again, so you take in a deep breath. And all you can smell is that old truck in front of you. The air you're breathing in isn't just bringing oxygen into your lungs, it's also bringing in those car fumes and everything else in the air around you. And whether you're sitting in traffic, or you live, or work, or go to school near a roadway, millions of us are breathing in traffic-related air pollution, or TRAP. TRAP refers to different air pollutants that come with being near roadways, coming from road dust or car emissions. And as you can imagine, breathing in this pollution can cause harm across the body and increase risk for disease. One of these diseases is Alzheimer's disease, which affects the brain to cause memory loss in 7 million Americans alone. And unfortunately, Alzheimer's is a disease that we don't fully understand yet, and we don't really have a cure for. We know that breathing in TRAP increases your risk for Alzheimer's, but we don't know how. How is it that breathing in pollution somehow affects your brain and causes memory loss? What we do know is that TRAP is critically linked to inflammation. Inflammation is your body's immune system responding to something foreign in the body, like air pollution, or to injury or disease. You can picture it as the redness and the swelling that you get in your thumb when you have a cut or a splinter, for example. And in the short term, inflammation is a crucial way for your body's immune system to protect the body. But long-term sustained inflammation, which is what we see with TRAP exposure, can overwhelm cells And this increase in inflammation in the brain is a key driver of Alzheimer's disease. So since we know that breathing in TRAP increases inflammation in the lungs, and inflammation in the brain increases Alzheimer's, we can link together the lungs and the brain and air pollution and Alzheimer's through inflammation. And that's exactly what I'm doing in my research. I'm proposing a model in which breathing in TRAP, these foreign particles, increases molecules that signal inflammation in the lungs. These can then spread throughout the body and reaching the brain, increase inflammation there to promote Alzheimer's disease. And how am I testing this model? Well, I'm using rats that are exposed to air pollution. And these rats already show increased markers of Alzheimer's disease, again, underscoring the relationship between the two. I'm then giving them a drug that stops these molecules that signal inflammation to see whether by decreasing inflammation, we can decrease Alzheimer's markers with air pollution exposure. By looking at the role that inflammation plays in Alzheimer's, we can better understand different ways to potentially treat the disease. And by understanding how TRAP drives Alzheimer's, we can better understand the mechanisms through which air pollution affects our bodies, meaning we can help guide regulation to prevent exposure to TRAP, or come up with preventative treatments for those of us who are already breathing in TRAP on a daily basis to reduce our risk for getting Alzheimer's in the future. So the next time you're stuck on the freeway, take in a deep breath, but remember, your brain health is being shaped by every breath you take. Thank you.
Could you tell us what inspired you to pursue this research topic?
Sure. I was always really interested in kind of how the brain breaks down, which is neurodegenerative diseases like Alzheimer's, Parkinson's, those sort of diseases. And that's what I was doing before grad school. And once I started, I found my current PI, Dr. Pam Lyons' lab, and I'd never really thought about the environmental stuff that happens and how that kind of affects diseases like Alzheimer's. And as soon as I saw these features, I was like instantly drawn to it. So that's kind of how I got into it.
I'm wondering, your last statement about when you're sitting on the freeway, does it make a difference whether you're in a car or on a motorcycle asking for a friend?
I get that question a lot, actually, and people always ask me, should I be driving with my windows up? And the irony is, I was practicing this whole talk in my car with the windows down, so I don't know if I'm the most qualified person to say it, but yes, there's definitely, like, having a filter definitely helps, but also we don't know a lot about TRAP composition yet, so there's a different range of sizes, so there's obviously differences that you get from different types of TRAP components. But yes, as much as you can decrease your exposure, the better.
Yeah, in fact, you mentioned sizes. I was wondering about that when so multiple ranges,
yes,
right?
So you have, not to get too technical, but you can have like super small nano-sized particles. You can have ultrafine, those are like the smaller nano-sized particles, and you can have larger particles. The most common one that we see with TRAP or even wildfires is called PM2.5, which is particles that are 2.5 microns wide. So they're still really small for us, but you have particles that are like 1/1000th of a human hair. So obviously, like, there's not a lot of filters or stuff that are great at capturing that, but yeah.
Right. What's the most, take us along with you in your research, right? What's the most important, interesting part of your day-to-day research?
My most interesting part is that we actually had talked about rats that are exposed to air pollution. We have a vivarium, which is where you house your animals, right next to a freeway. So we have a bunch of rats that are next to the freeway that are directly breathing in TRAP through a traffic tunnel. So the most interesting part of my week is going there once a week with one of my lab mates, Aish. And we go there and we like change cages and everything. So yeah, that's part of the science that's really important, but it's not doing like lab work.
Right. Thank you so much.
Thank you.
Our next speaker is Christian Pratt.
I don't have a slide.
Oh, even better.
Yeah. Laptops, smartphones, and data centers are expected to consume up to 20% of all global power generation by 2030. In fact, all of our current technology dissipates at least 10,000 times more heat into the environment than what's theoretically possible. One cause of this energy waste comes from electrons encountering resistance as they're conducting electricity, releasing waste heat in the environment. This is why you feel your phone getting hot when you're doom scrolling at 2 a.m. One way to address this is to build an alternative computing device that can be cooled down to extremely low temperatures, like -450 degrees Fahrenheit or -268 degrees Celsius. at these mind-numbing temperatures, electrons will no longer face resistance as they're conducting electricity, generating only a fraction of the wasted energy. But I don't want to work at negative 450 degrees. Instead, my research develops mathematical methods and simulation tools for building these energy-efficient alternative computing devices. I showed that these devices could produce around 10,000 times less heat than our current technology when performing the same computations. To give you an idea of this scale, imagine that we were able to shrink an elephant to the size of a mouse. Eventually, when these devices are built, they will live in a storage apparatus. And even though this technology is still in its infancy, it will live in a storage apparatus. And even though this technology is still in its infancy, it will only account for 20% of the entire apparatus's power consumption. And everything else comes from things like external monitors and wiring. My research could help play a pivotal role in keeping our future power consumption down while minimizing our energy waste, all while conserving costly research resources. That way, we can all continue doing everything from doom scrolling on our phones to performing cutting-edge research. Thank you.
What inspired you to participate in Grad Slam?
Yes, what inspired me to participate in Grad Slam was I value when scientists are able to communicate to the general public about their research. And when I was first getting interested in physics, one of the people that I looked up to was Carl Sagan. And one of my favorite books of all time is The Demon Haunted World, where he talks about how to communicate science to any person of any background in all kinds of relatable ways to them. And for me, Grad Slam is one way for me to step in these popularizer shoes and to really try to hone that skill of talking to people. Yeah.
What about this specific line of research? What led you to that?
The specific line of research was, I was always interested in computers, and then physics kind of stole my heart. And eventually, I ended up finding the physics of computation, which is why I really wanted to get involved in it. And I think it's a really big deal that now that AI is becoming this huge boom, we need to take care of our data centers and make sure that they're not consuming so much energy.
Right. I mean, what you're describing is a major paradigm shift, right?
Yeah.
When do we get one?
Yeah, my research is mainly about, it's a prototype device. And yeah, not currently on the market yet, but yeah.
We spoke yesterday during the poster session, and so I got to see more visual details about what you were describing. Can you give us a sense, you were talking about data center, et cetera, a sense of the scale of the device, if we can call it a device, of the unit?
Yeah. So it's on the scale of like micrometers, and people are able to build these kinds of chips that operate at these really low temperatures. They can they have them in chips currently, but not at the point where it's like we have our phones, for example.
Yeah. Okay. All right. Thank you so much.
Thank you.
Next speaker is Olivia Feldman.
When most folks in this room hear Patagonia, they might picture towering snowy peaks, like the logo on the popular American clothing brand. But on the Argentine side, most of Patagonia is actually a patchwork of dry grasses, shrubs, and rocky canyons that are home to wild animals like guanacos, small wildcats, foxes, and pumas that you can see on the left side of the slide here. On the right side of the slide, however, we see the most common animal in Patagonia, sheep. lots and lots of sheep, and sheep ranchers, Argentine Patagonia's gauchos. All of these photos were collected by my camera traps that were placed all at the same field site, a working sheep ranch. In fact, over 90% of Argentine Patagonia is private land, most of it used for sheep ranching. This means most wildlife lives on ranch land. But sharing space can be complicated, especially when it comes to ranchers, sheep, and wild carnivores. You see, sometimes wild carnivores hunt sheep. This is a big loss of income for ranchers, so ranchers kill sheep in order to prevent this. Problem is, we need wild carnivores on the landscape to hunt wild prey so that there's enough vegetation to maintain a biodiverse ecosystem. And that is good for sheep. So, how can ranchers keep sheep safe without having to kill wild predators? Well, turns out this form of human-wildlife conflict is common all over the world. And there are things to try, but nothing is one size fits all. And what may work for a cattle rancher in Montana or a goat herder in Nepal may not work for a sheep rancher in Patagonia. And trying things out one by one is expensive and not financially feasible for most ranchers. This is where my research comes in. In southeastern Argentina, I get diagnostic. And I'm identifying the factors that make a wild carnivore more likely to hunt sheep on a given ranch. To do this, I combine tools from wildlife biology and social science to build a complete picture of how ranchers, sheep, and wild carnivores interact. I complement the long hours spent setting up camera traps to study wild carnivores with long evenings spent sipping mate and interviewing ranchers as they walk me through what they're seeing on their land. Interdisciplinary approaches like this are key because they prioritize information from people the same way we do data about wildlife and the environment. This is not common in my field, but it is integral. Not only does this give wildlife biologists a clearer picture of the conflict to better work with ranchers, but it also means that the people who currently live this challenge are an active part of its solution. Thank you.
Tell us about your future career goals.
Oh, God. So I'm kind of trying to shape something together. In an ideal future, I'm based in Argentina, running research projects that actively inform human-wildlife conflict prevention, hopefully on a number of ranches.
I'm interested in these cameras that are set on private land, and I'm wondering about the difficulty it is to get. I mean, you talked a little bit about communicating with the people there, but I'd like to hear more about that.
So I bridge a couple of worlds here, right? I kind of have my wildlife biologist hat on sometimes, and I collaborate with an NGO on that front. So I also have camera traps set up on a reserve that used to be a sheep ranch. And then I also work really hard to integrate into the local community that I work in. So I have visited every rancher that I've interviewed. I've visited them basically twice at least, usually to first come over, say hi, introduce myself, talk a little bit, and then if they're comfortable, we run the actual interview. Now, Argentines are really social, so this often means, like 2 to 3 hour visits each time. Lots and lots of mate. But yeah, the rancher here that's permitted me to put camera traps, and all the pictures come from that ranch, we've been working together for about 3 years. And he's not necessarily an avid conservationist, but the trick to a lot of the ways, the trick I think to working with people is really understanding that people are just trying to deal with their problems. And this happens to be an environmental one as well.
Okay. What other hobbies do you have? Well, what hobbies do you have besides ecology and mate?
I used to have more hobbies before grad school, but that's probably most of us. When I'm in Argentina, so my partner lives in Argentina, we like to check out new sites, trace new roads in abandoned places, and look for fossils. There's a lot of fossilized shark teeth in this random little oil city that he lives in.
Right. Thank you so much. Our next speaker is Nana Moraes.
Who here has a dog? I know, it's your baby. I don't need to see pictures, but tell me, do they always behave? You've probably experienced this situation. You go to a friend's house where the dog is barking, jumping, even trying to bite someone, and the owner doesn't know what to do. This person was me. In 2012, I rescued Louis in Brazil. Over time, he became aggressive, just like other animals I'd had before. Then I realized that the problem was not them. The problem was me. And in fact, I was always angry, irritated, anxious. Then I started to watch The Dog Whisperer, the TV show with Cesar Millan, you know? He talks a lot about energy. And I already knew energy from physics and biology, but this was different. That show led me to study social cognition, our ability to read and respond emotions, along communication, behavior, and anthropology. When I say energy, I mean emotions. It's the presence we carry in our body, in our voice, and the animals feel that instantly. Let me give you a visual example. When I walked Louis, I held the leash tightly, shoulders tense, and Louis reacted, pulling, barking. When I relaxed and became calm and confident, Louis changed too. This is energy. And this applies to other social animals like horses, cows, pigs. That's why I am doing an ethnographic research. That means I go into field, animal rehabilitation center, and I observe how reactive animals change over time. I record interactions, interview professionals, and I explore the techniques they are using there. And why does it matter? Because my research treats energy as a lived relational force, not as a metaphor, and shows that humanities are the foundation of other sciences. In veterinary care, for example, understand this can reduce sedation, accident, cost, retain, and shows that my research is a reminder that we often call animal behavior might be a conversation we've never learned to hear. Thank you.
Thank you. I have an Australian Shepherd, so we need to talk after this. What surprised you the most in your research?
It's weird because I am a lawyer in Brazil. And here I am doing the PhD in anthropology. And I really started to study social cognition, anthropology, human and animals relationship because of Louis. But I am animal protector in Brazil, and I rescued more than 100 animals. Not only cats and dogs, that's the most common, but even a penguin, and we don't have penguin in Brazil. And I saw that the animals that lived with me were a little agitated, so I realized that the problem was not all the animals in the world. Probably I was passing my energy to them.
Yeah, that makes sense. Is there a skill that you'd love to learn that you don't have?
A skill?
A skill that you'd love to learn?
Yes. I talked a little about Cesar Millan, and I know it's the dog whisperer. I don't know if you know him. But other behaviorists are using this technique to become calm and confident. I am a little anxious, as you can see, and I am trying to learn the technique they are using to put in practice in the field, like to become calm in a moment, like to put your mental state in a good way.
What's next for your research?
The next, first I will do my qualifies soon. But after that, I will go to the Dog Psychology Center in Santa Clarita, and in this place they are doing this study of energy with animals, with a lot of animals, rescued animals, like big and small, like even chickens, they are using that. And my goal is to work there with them. That's my...
Well, that's a great goal. Thank you so much.
Thank you.
The next speaker is Sarah Lee.
Thank you. Have you ever thought that your food intake and vitamin supply can affect your health? Of course you know that, but have you ever thought that it can affect your future kids' health? Well, it does. Here's an interesting puzzle. In 1998, the US started adding folic acid vitamin to food on top of the prenatal vitamins recommended during pregnancy to prevent serious birth defect that worked well in decreasing the birth defects. But also it has led to increase in the folate level in some pregnant women. 2 years later, we started seeing more and more children being diagnosed with autism. While genetic factors can explain some causes, still 40% of autistic cases still unexplained, and we can appreciate the increase of autism in families, classroom, and our communities. So the question is, could environmental factors like folic acid be involved? Interestingly, a global epidemiological study in Boston found that autism in kids was increased when their moms took too high or too little folic acid during a pregnancy. So to solve this puzzle, I needed a way to study how baby brains develop under effect of folic acid imbalance, but I couldn't look inside pregnant woman's womb, and animal models fall short. Then, here's the surprise. We can grow human mini brains in the lab, and this is exactly what I did. I grew teeny version of baby brains in our lab. They are made from human cells, and they grow and develop just like real brains do during pregnancy, regarding both the structure and the electrical activity that matches actual developing fetus brains. These patterns, under normal balanced conditions during pregnancy, form countless connections needed for everything for brain function, from basic function to complex thinking. What we discovered is that when these mini brains got either too little or too much folic acid, the brain cells, we call them neurons, couldn't talk to each other properly. Imagine a classroom when everyone whispers instead of speaking normally. That's what happened in both cases. By using special equipment that can hear neurons talking, we found that both extremes just quite the neurons. What was striking is that this disrupted pattern closely mimic what scientists have observed in kids with autism, less complex brain activity compared to typically developing children. This tells us something important. Just like Goldilocks looking for the perfect bowl of porridge, developing a brain needs just the right amount of folic acid. My research could help inform better prenatal guidelines, even personalized for prenatal nutrition, because when it comes to building the next generation of brains, precision matters. Thank you.
We can grow mini brains in our labs.
Yeah, and they are human.
Can you take us through your path? What led you to this field and then specifically to this research?
Of course. So first, I'm a physician. I'm a pathologist taking my MD degree. And then I noticed how much the autism prevalence increased even in families. So that was like heartbreaking and making a lot of burden on families. And we can appreciate that it's growing so fast, while genetic mutations can't explain this high auscultation. So, and no mention of the environmental factors, so it seems that there's one factor that can play a role here. So I got very interested in the idea and the work, but also animal models are totally different from human brains, especially regarding the complexity and so on. So I was impressed that we can mimic what's going on inside the developing human fetal brain regarding the complexity, brain layers, and all of this to close the gap as much as possible and see how this can be influence the folic acid imbalance during pregnancy.
No, how did you get to UC Davis?
What
led you to join here?
Yeah, so UC Davis, I appreciate so much the diversity here, and regarding the students and also the different departments, ideas and all of that, and also the weather is extremely nice, so it's a big advantage to me. And also I like so much the lab that I joined because the idea was done at that moment on the mice model, but then there was a lot of shortfalls in the mice brains and so on. So I'm so impressed that we can now have this technology that we can so much mimic the human brains in this area, because of course studying anything during pregnancy is ethically and impossible kind of. So I find that this is I hope that it can help the public health awareness regarding this problem. Because while can I mention something? Okay, while the WHO recommended upper level of folic acid intake during a pregnancy that shouldn't exceed 1,000 micrograms, there is no increased awareness. There is no follow-up guidelines on this. It's just something okay, it's too much of a good thing, there's nothing will happen. But actually no, because the folic acid introduced in the fortification and the supplementation, it's fortified by industry and it's oxidized form, totally different from the reduced form naturally present in food, because it needs first to be reduced to be utilized by our cells. And the reducing enzyme, very slow in humans and highly competitive. So you can end up having during pregnancy high amount of folic acid that you can't guarantee that you will utilize it by your body. And then you will have high unmetabolized folic acid that come in accordance with a study from autistic cord blood that will show high amount of unmetabolized folic acid. So there's a lot of correlations between the subjects. So I feel, hopefully, I'm talking a lot. I'm sorry.
Passionate about it. Thank you so much.
Thank you. I'm not going to questions.
Yerika team is our next speaker.
Imagine your brain as a vast city made up of a complex network of roads, highways, bridges, connecting all of the different areas. These different pathways allow for memories and thoughts and emotions to travel seamlessly, kind of like cars would while navigating through a well-organized traffic system. But now for a moment, let's imagine these roads begin to crumble. Bridges collapse, highways shut down. Suddenly it seems like the city's entire communication grid is in chaos. This is what happens in the brain when brain cells and their connections break down, a process that we see in many mental health disorders like depression, anxiety, and addiction. And when we look at the current treatments available for these kind of conditions, we see that they primarily focus on managing the symptoms, but they're not really addressing this underlying structural breakdown itself. What's more is that these treatments often leave patients with a long list of unwanted side effects, and they leave key aspects of the conditions unaddressed. So what if, instead of just easing these traffic delays, what if we could actually rebuild the roads? What if we could restore the bridges, reinforce the highways, and restore the brain's structural integrity in a way that fundamentally modifies the very nature of the disease itself? And what if we could do that with just a single drug at a single dose? Researchers in the Olson lab at the UC Davis Institute for Psychedelics and Neurotherapeutics have found that psychedelic drugs like LSD and DMT can both rapidly and persistently restore these lost connections in the brain. And what's really critical here is that this restorative effect has been linked to the long-lasting improvements in mood and behavior that we know these drugs cause. Now, while these drugs are notorious for some hallucinogenic side effects, our lab has actually found that you can tease apart these restorative qualities from the hallucinations and make new psychedelic-like drugs that only harness these restorative qualities. So as the field pivots towards synthesizing and identifying these new psychedelic-like drugs, we need better tools to get us there faster. And that's where my research comes in. As a member of the Olson lab, I'm developing an automated tool to allow us to find the best drugs possible for restoring these lost connections. Essentially, a tool that will allow us to find the best materials needed to rebuild the brain's roads and bridges. Using an advanced imaging technique, I take pictures of thousands of brain cells treated with our potential drug candidates, and then I assess the effect on cellular structure using custom-built software from within our lab. This allows us to pinpoint exactly which drugs are capable of regrowing cells and make new treatments that don't just address the symptoms, but are actually rebuilding the road to recovery. Thank you.
Thank you. Is it an issue if you don't have the hallucination aspect?
Yeah, I mean, this is the
treatment?
biggest controversy in the field right now. People for, tens of years, centuries, have wanted to take these psychedelic compounds for this hallucinogenic experience, for the mystical experience, the emotional breakthrough. These can be really powerful for people. But what's really interesting to us is long after the hallucination is over, long after the drug leaves your body, people still report feeling better. There are these long-lasting improvements to their moods, a so-called afterglow effect. So the question is why? Like, what's causing that? That's what our lab is interested in, and more research from our lab is pointing to the fact that it's this restorative quality in the drugs that's actually promoting these long-lasting changes. So it's not that we don't know if the hallucinations cause a benefit or not. They likely do, they could, but they're not the whole story.
Right.
And if we can harness one aspect of the story and make safer, more accessible, more scalable treatments for a larger patient population that maybe can't tolerate a psychedelic experience, we have more treatments for more people.
Right. And would that help with the regulatory aspects of it?
Absolutely, absolutely. Accessibility is a big thing right now. So I mean, there's definitely psychedelic-assisted therapy that is being implemented in the clinic, drugs with the hallucinogenic effects being used, but can you imagine the insurance hurdles? Getting the approval, and it's definitely only specific patient populations that will have the privilege and the luxury of getting these kind of treatments. So if we can make it more accessible to different people, then people can take them at home and we'll be able to overcome these kind of hurdles.
Okay. What's next for your research?
Well, using my tool, hopefully we'll be able to find some of these drug candidates, and then the next step is to dig further. What are they doing on a cellular level? What are they doing on a brain-wide level? And then, is it actually relevant for the treatment of depression, for post-traumatic stress disorder? So those are kind of the next questions that we're going to answer once we can identify drug candidates from the automated tool that I'm developing in the lab.
All right. Thank you.
Thank you.
Next speaker is Jacqueline Lang.
Imagine this. You're walking next to a stream up near Seattle, and it starts to rain. Suddenly, you start to see thousands of salmon float to the surface and die. Well, this exact scenario had been baffling researchers for decades, until 4 years ago when they discovered the culprit, a chemical in tires called 6-ppd-quinone. When we drive, tiny rubber particles shed from our tires and end up in waterways where they can release toxic chemicals. So we were curious, could 6-ppd-quinone be causing harm to our native salmon here in California? Salmon are a keystone species, which means that a decline in their population can ripple throughout food webs. They support a $1.4 billion fishing industry here in California, which provides 23,000 jobs. They also hold deep cultural significance to our indigenous communities and our fishing hobbyists. But these populations are still plummeting due to issues like climate change, habitat loss, and pollution. In fact, for the last 2 years, our salmon fisheries have been closed due to such low numbers. So, to figure out how toxic 6-ppd-quinone is to our salmon, I exposed young steelhead and Chinook salmon to a wide range of concentrations. This allowed me to predict how many of our salmon are at risk of dying due to real-world pollution levels. For steelhead, it was concerning. About 7% of steelhead are expected to die within 4 days of exposure to 6-ppd-quinone at levels that are already present in the San Francisco Bay. However, on the other hand, our Chinook salmon were completely unaffected, even at concentrations 10,000 times higher than what they would ever experience in the wild. These differences in survival suggest that these two species are processing 6-ppd-quinone very differently. But survival doesn't tell the whole story. Even at very low real-world concentrations, fish from both of these species were behaving differently. Some of them would slow down and hide more, while others would dart around erratically. These changes could suggest that 6-ppd-quinone is affecting their brains and how they're able to cope with different issues that they come across in their environment. These slight changes can really affect populations even if it doesn't cause death. These issues are revealing that we are seeing both resilience and risk in the wild. Some salmon may be able to survive tire pollution, while others may not be able to, and that's a real big concern for us. And so, by understanding these issues, we can start coming up with solutions like better tire formulations and stormwater management to keep our streams clean and our salmon healthy and strong for generations. Thank you.
How did you get interested in that?
That's a really good question. So I've been an Aggie for a long time. I also did my undergrad research here. And when it was in my last year, I got to spend an entire summer at the UC Davis Bodega Marine Lab, which was one of the greatest experiences I've ever had. I got to work with a lot of really cool organisms like purple sea urchins and squishy worms and jellyfish. And at that point, I was stuck. There was nothing else that I was going to be able to do other than aquatic toxicology.
Yeah. Not a bad place to work, Bodega, right?
Yeah, it's beautiful.
Are there any other species that, I don't know if you've looked into this, but that might be sensitive to this same chemical?
Yeah, so I have looked at other species. I've looked at 2 species of smelt that are highly endangered here in California. And they're both similar to the Chinook, where you can basically dump 6 ppd quinone into their tank and they are doing just fine. But actually other researchers have looked at other fish species and even aquatic invertebrates. And the only fish who seem to be very sensitive are salmon. And like I mentioned, only some salmon. And we still don't really fully understand why. A lot of researchers are really looking into it right now, so hopefully we'll know soon.
Okay. Do you have a favorite animal that you worked with?
I've worked with a lot of really amazing animals, and my favorite is one that might be a little surprising. They're called freshwater mussels. They're not very cool to look at, but they kind of look like just like rocks, essentially. But they have a really cool reproductive biology that is just unlike any other animal I have ever met, and I'm obsessed with them. I think they're so cool.
It's like having a pet rock, I guess.
Yeah.
Where do you see yourself after graduation?
I've been thinking about working at the California state government. The California EPA has a lot of departments that work with different toxic chemicals, like the Department of Toxic Substances Control and Department of Pesticide Regulation. So I'm really interested in doing what's called risk assessment, which is where you really are just kind of reading all of the papers that everyone is publishing and synthesizing them into a succinct but very detailed document about when we're going to be hitting toxic thresholds that are concerning for either humans or wildlife.
All right, thank you so much.
Thank you.
Our next speaker is Diana Malarchik. I might as well just give you this.
We've all heard the saying, you are what you eat. But what if I told you that that's truer than you could ever think? What if I told you I could tell you all about your childhood diet, even how long you were breastfed, just by looking at one small thing, your tooth? When people think of archaeology, they often think of Indiana Jones dusting off some old bones in the desert somewhere. But the archaeology of today is full of cutting-edge technology. As an archaeologist, I study geochemical signatures, specifically nitrogen and carbon, from skeletal remains to learn all about what people were eating in the past. You see, teeth retain the chemical signatures at the time when they were grown, starting at birth and ending in your late teens. And teeth grow differently than the rest of your skeleton. Teeth, just like trees, grow in rings. They start at the top and grow down. And based on this anatomy, I can sample these growth rings and get nitrogen signatures and create dietary timelines for each individual. Looking at these early diets, I focus on the first few years of a person's life when they were breastfed. My work highlights major shifts in breastfeeding and maternal behavior in the past. And the people I study are from San Francisco, but they lived there in the 1800s. Looking at their early diets, I have found 3 patterns. So we see a typical pattern, gradual weaning, where an individual is slowly weaned off of breast milk over a few years. But then we see 2 new patterns, shortened weaning, where an individual only has about 6 months of breastfeeding, and an absent signature, which means that individual didn't get any breast milk at all. This is a significant discovery, as it means these people survived childhood with little or no support from breast milk. And before the 20th century, breastfeeding was the only way an individual could get safe food and immune support. Without this, mothers would have turned to early versions of formula, which was just soaking bread in water. And in a time before germ theory and boiling water, this would have introduced untold pathogens. At this time, there was a major shift in economics and a rise in capitalism, and this forced working mothers to make an impossible decision: earn a living or feed your baby. When comparing upper and lower classes, we see that absent and shortened signatures are significantly more prevalent in burials from public cemeteries, meaning that children from lower socioeconomic statuses were the most affected. This is not an issue of the past. In the US, only 13 states offer some sort of paid family leave, and it's often only 6 weeks. Working mothers today are still being forced to make the same decision that their great-great-great-grandmothers made in the 1800s. And unless we can learn from the past, we will be forced to relive it.
Thank you. Was there anything in particular that deepened your passion for this research topic, something that made it your research topic?
Not something, but someone. About 7 months ago, I gave birth to a little boy, Pedro. He's the greatest thing. And I felt like I always had this appreciation for my work, because we might just see these little dots and stuff, but when I look at this, I see parents. I see them striving to keep this child alive who had cholera, who had tuberculosis, who would scream in the middle of the night, which now I have firsthand knowledge of. And I see perseverance in the past. And so, yeah, now having a little kid myself, I look at this and I just see resilience in humanity, and I love it.
Well, that may be if it's too personal, I mean, if you tested your son's teeth, what would you find?
Okay, that's a good thing. So when I talk about when they're grown, we say, teeth grow at birth throughout your late teens. That's just your permanent teeth. So your deciduous teeth, your baby teeth, they grow when you're in the uterus, when you're still in the maternal diet. So his teeth right now reflect my diet. So it would just be pizza and pints and oranges. That's pretty much what it is.
Do you use any cutting-edge technique? I mean, you described quite a few types of techniques.
Yeah. This is actually only a third of my dissertation. Yeah. I have a very big project. So I also work in the environmental toxicology lab with Dr. Parker, and we look at proteins in dental enamel, and we can isolate specific amino acids and look for one amino acid change that's located on the Y chromosome. So I can take your tooth and tell you if you're a male or a female just by looking at a protein.
Right.
And I also look at heavy metal exposure in the past. So that is also laid down in dental enamel. So we have a whole side project where we look at lead exposure, arsenic, mercury, which if anyone here reads the new Consumer Report about baby formula, is also not a problem in the past. So a lot of my work, unfortunately, is still really relevant.
Right. Well, thank you so much.
Thank you.
Our next speaker is Jordan Crowley.
All right, thanks. So imagine you're on the bus, and you overhear behind you a language that you don't recognize. As you listen, you might think, wow, that language sounds beautiful, or perhaps harsh and angry, or even, wow, that language sounds really complicated. But what makes someone who has no prior experience think of a language as being more difficult? That's exactly what fascinates me and is what my research is all about. You see, languages aren't just single entities. They come in varieties, and dialects can differ dramatically, even within the same country. China, for example, is home to many dialects, some as distinct from each other as English is to Dutch. My research looks at how non-Chinese speakers perceive the difficulty of different Chinese dialects. Participants with no significant exposure to Chinese listened to recordings of Chinese dialects and ranked them according to how difficult they sounded. Their responses provided some interesting results. for example, one of these results was that these listeners thought that the Beijing dialect, which is based on Standard Mandarin, was the easiest. Meanwhile, a rare dialect from Wenzhou was considered the hardest. But why? The answer likely lies in exposure. You see, Mandarin is the most widely used Chinese dialect in the world. It's used in media and technology all over the globe. Even if you've never studied Chinese, it's very likely that you've at least heard Mandarin in passing. On the other hand, dialects like those from Wenzhou remain very much unknown by non-native speakers, making them feel more foreign and therefore more difficult. So this has huge implications for language learning. If our perception of difficulty is based on what we're exposed to, then our ability to learn a language, or more importantly here, our willingness to even try to learn that language, is being affected before we even begin. This can also highlight hidden biases in how we approach unfamiliar cultures. Should a language be considered difficult or more hard than another, does that discourage us from engaging with that dialect and that community at all? Do speakers of these dialects face greater linguistic barriers in their world? And in a world that is increasingly interconnected, yet more divided than ever, can these biases reinforce those cultural divisions that are already present? You see, by studying this intersection of language difficulty and dialects, we can better understand not just how people hear language, but how their perceptions can be affected by real-world language learning and cultural attitudes. So, next time that you're on the bus, be sure to think of me and my research, and be sure to keep one ear open when you're on your route. Thank you.
Thank you. How did you get interested in linguistics? I mean, it's a fascinating field, but how did you personally?
It was an accident, actually. So I started undergrad as a pre-med major. I was one of those, I'm going to med school, nothing's gonna stop me. And then I took organic chemistry, and I was like, oh, I don't know if I want to do this forever. So, I mean, I still like the sciences, and I like chemistry and biology. But after I took OChem, I was like, I need, I think, another path. So I just started taking a couple random GE classes. I took a psych class, I took a geography class, and then I took a linguistics class. And it made me realize, why was I not doing this forever? Because I've always had an interest in language and geography and traveling and international relations, and linguistics was really the perfect opportunity for me to find some sort of mesh between all of those things.
Okay. You mentioned how the perception of difficulty or ease of a particular language changes the intention to learn it, right? So people may be reluctant. Is there, can it work both ways? So do people sometimes think, oh, that sounds really difficult, I'd love to study that?
Probably. I'm sure there are people who are motivated by that. So does anyone in here speak Chinese? Any dialect? Great, some of us. Most of us probably don't. So if I asked and polled specific people, would you be willing to learn Chinese? I would imagine the majority of them are probably, no, that might seem kind of hard, or maybe like, I'm already in my 40s, I don't need to learn another language, which is a myth, by the way. You can learn a language. But I'm sure there's a few handful of people in here that might be motivated in the opposite direction.
Right, right. I'm wondering about something that's connected to this. It's the perception of accents.
Okay, sure. What about it?
Trick question. Well, I'm wondering if it's relative, right? So if, you know what I mean? If I hear an accent as a native French speaker, does somebody who's a native Chinese speaker, Mandarin speaker, would hear a different accent?
It's possible. That's not something that I tested for in this particular study, but one of the things that I did have to take into consideration was, even though the people who were speaking my dialect spoke their version of the dialect, I didn't really have a way to control for their particular accent within that dialect. So it's probably in the future going to cause a little bit of problems in how I do my data analysis. But I'm not certain exactly the broader scope that individual accents would have on it.
Okay. Well, thank you.
Thank you.
All right, well, thank you all for the wonderful presentations. We will now open the voting for the People's Choice Award. All of you here in person can scan the QR code right here to access the survey tool. Our virtual attendees, please click on the shared link that's in the chat box, and you'll be able to access the survey there. Voting will be open for 15 minutes. At the same time, our judges will convene to determine our winners. As all of this is happening, and once those of you here cast your vote, please feel free to enjoy some refreshment and mingle in the Commons room right here. We'll meet back here in 15 minutes to announce the 2025 prize recipient. Thank you. All right, welcome back, everyone. This time it's for real. I hope you had a chance to enjoy the refreshments. Before we move forward with the award portions of today's program, I would like to thank again our judges, contestants, and staff volunteers. I would also like to, no, I want to go back to that. There you go. I would also like to give one last thank you to our sponsors. Without you, this event wouldn't be possible. We'll begin by announcing the winner of the Public Impact Prize. This prize is awarded by Public Scholarship and Engagement and recognizes outstanding research that has or will have significant impact by addressing a public concern or topic. Will the winner please come join me on stage once their name is called to receive their trophy? Our Public Impact Prize goes to Emma Bernard. Thank you. The next prize is the People's Choice Award. This prize was selected based on audience input. I was telling just moments ago, you all voted, I'm sure. Will the winner please come and join me on stage next to the podium? And to receive their trophy, our People's Choice Award goes to Nana Moraes. Now we come to the first, second, and first place prizes. I'm making sure I have enough envelopes. And the recipient, please join me on, next to the podium to receive your trophy when your name is called. Our third place prize of $1,000 goes to Olivia Feldman. Our second place prize of $2,000 goes to Diana Malarchik. Congratulations.
Thank you.
And now for our 2025 UC Davis Grad Slam winner. Oh, there you go. The first place recipient will receive $5,000 award and represent UC Davis on April 29th at the University of California 2025 Grad Slam Finals event. Congratulations to Yara Khatib. No. I was going to give. And wait, we also have. And wait, you get a certificate.
Thank you.
Congratulations.
Thank you so much.
In addition to this prize money, like I said, Yara will get to compete in the 2025 University of California Grad Slam Prize. Your name and your college name will also go on the permanent display. We have a plaque up there in the lobby. You can come visit it, you know. Congratulations again on these accomplishments, and I hope that you all join us, that everybody who joined us today will tune in for the April 29th UC Grad Slam. There will be a live stream for that and to support Yara. Another big thank you to all our top 10 finalists today and every UC Davis graduate student who participated in the competition, and thank you again for being here today. I invite you to stick around and join us for the other event reception.